results. I'd like to take the opportunity to mention that this study has been released as a preprint. Details are at the bottom of each slide. Ovarian tissue cryopreservation is used to preserve fertility in girls and women undergoing gonadotoxic chemotherapy treatments. The method involves the removal of an ovary or ovarian biopsy that is then dissected and cryopreserved with the aim of preserving the primordial follicle pool contained within the ovarian cortex. Ovarian tissue cryopreservation is becoming increasingly well established worldwide. However, it requires specialist equipment and dedicated facilities. Accessibility to this method could be improved by delaying processing and cryopreservation of the ovary for several hours. This could allow for the tissue to be transported to a central cryopreservation facility and facilitate coordination and workload management at the site. A delay between ovarian collection and processing could also be necessary in the field of conservation, where animals from endangered species may be collected far from laboratory facilities. Delayed processing and cryopreservation for up to 20 hours at 4 degrees Celsius has been performed clinically, with one subsequent live birth reported. However, the impact of such a delay on the health of the primordial follicle pool remains controversial, with studies reporting either no effect on follicle density and health or a negative effect on both. In this context, the aim of this study was to determine whether a processing delay of 24 or 48 hours had an effect on primordial follicle morphology using fresh, cryopreserved and xenotransplanted ovarian tissue using sheep ovaries as a model for human. Ovaries from female lambs were obtained from a local abattoir and transported on ice in L15 medium. Once at the laboratory, one ovary from each pair was immediately processed, while the other ovary was stored at 4 degrees Celsius in transport medium for either 24 or 48 hours. At each time point, the ovaries were processed for cryopreservation using clinically relevant protocols, and the ovarian cortex was cut into strips approximately 5 by 2 by 1 millimeter. At this point, cortical strips were either fixed directly or cryopreserved. For cryopreservation, cortical strips are placed into cryoprotectant solution containing 1.5 molar ethylene glycol, 0.1 molar sucrose, and 3 milligrams per ml bovine serum albumin, and cryopreserved using controlled rate freezing with manual seeding. Samples are then thawed and either fixed or transplanted subcutaneously into skid mice. Xenotransplanted samples were retrieved after 10 days and fixed. Samples were fixed in buin solution, sectioned, and stained with hematoxylin and eosin. Primordial follicles were identified based on the presence of a single oocyte surrounded by a layer of flattened pregranulosa cells. Up to 50 primordial follicles were assessed in each sample for thawed or cryopreserved samples. Xenotransplanted samples were sectioned to completion and all follicles were analyzed. For these samples, follicles were further classified as primordial or growing. Samples were analyzed in a blinded manner by 2 researchers with some overlap to ensure concordance and agreement in the analysis. Primordial follicle health was assessed based on morphology using well-defined criteria based on a previous study from our group. Follicles were classified as being normal or having one or more of the following morphological markers: shrunken ooplasm, dense chromatin, eosinophilic ooplasm, and/or pycnotic nuclei. Based on these scores, follicles were graded as being healthy, affected, degenerating, or atretic. Statistical analysis was performed using a proportional odds model, and a logistic regression model. In both models, we adjusted for individual sheep as a random effect. I'll start by going over the results for the fresh tissue samples. Here you can see representative images of the tissue after immediate processing, a 24-hour delay, and a 48-hour delay in processing. The lower panels show the results of our analysis. Panel B shows the proportion of follicles for each health grade, combined for all animals. Follicle numbers are shown at the bottom of each bar, and panel C shows the distribution for individual animals. When we analyze these follicles, we could see that, as expected, the vast majority of follicles in fresh tissue were morphologically healthy, represented by the white columns, with a small proportion being classed as affected, represented by the light grey columns. We observed a higher proportion of affected follicles after a 48-hour processing delay. To analyze this data, we used a proportional odds model to determine whether a delay in processing affected the likelihood of primordial follicles being classed as healthy versus affected, degenerating, or atretic, as shown in panel D. The error bars represent 95% confidence intervals, and if they don't cross 1, that represents a statistically significant result. So you can see here that a 24-hour delay in processing had no effect on primordial follicle morphology, whereas a delay of 48 hours resulted in follicles being 2.5 times more likely to be graded as affected, degenerating, or atretic. So based on this, it seems that a 48-hour delay in processing has an adverse effect on the health of primordial follicles in fresh tissue, while a 24-hour delay had no effect. Next, let's take a look at the cryopreserved tissue. What we can immediately see here is that the follicles in the cryopreserved tissue look less healthy than their fresh counterparts, which is consistent with the literature. When we looked at these groups more closely, we could see that similar to the fresh tissue, a 24-hour processing delay had no effect on primordial follicle health. However, interestingly, after a 48-hour delay in processing, there was a higher proportion of healthy primordial follicles, translating to a 44% decrease in the odds of follicles being graded as affected, degenerating, or atretic compared to the immediately processed tissue. So to summarize the findings for the cryopreserved tissue, a 24-hour delay in processing had no effect on primordial follicle health, whereas follicles were actually healthier after a 48-hour delay in processing. The final aim of our study was to look at the functionality of this tissue. So we assessed the cryopreserved tissue after a 10-day xenotransplantation into SCID mice. All grafts were retrieved after xenotransplantation. However, the number of follicles in each graft varied substantially, with some grafts containing no follicles. Interestingly, only the 48-hour delay group contained follicles in all grafts. The primordial follicles that did survive transplantation were overall quite healthy, and there was no difference in follicle health between the three groups. We also looked at follicle development in these samples and found that they contained a high proportion of growing follicles, indicating that the follicles were functional and capable of growth. There was no difference in the proportion of primordial versus growing follicles across the three groups. So to summarize the last portion of the study, delayed processing and cryopreservation had no effect on follicle health following 10 days xenotransplantation. In conclusion, a processing delay of 24 hours at 4 degrees Celsius did not impact primordial follicle health in fresh or cryopreserved tissue. This is consistent with other studies using human ovarian tissue. Furthermore, we found that a processing delay of 48 hours had a negative effect on primordial follicle health in fresh ovarian tissue, but conversely a positive impact in cryopreserved tissue. This would suggest a potential role for extended pre-cooling of ovarian tissue prior to processing and cryopreservation. This might help to acclimatize the tissue, preparing it to withstand the challenges of cryopreservation and reducing the insult on the primordial follicle pool. However, this would need to be confirmed using human ovaries. It should also be noted that this study, the ovaries were left intact until the point of processing, so it cannot be concluded that ovarian biopsies would have a similar response. Xenografting of cryopreserved ovarian tissue confirmed tissue function in all experimental groups, with delayed processing having no impact on the health of primordial follicles or the proportion of growing follicles. Overall, these results demonstrate that ovaries can be stored at 4 degrees Celsius for up to 48 hours before cryopreservation, with no net negative effect on primordial follicle health. The findings are of high relevance to female fertility preservation, and in particular the way ovarian tissue cryopreservation is organized on a local and national level. An extended window for tissue processing would ensure that this technique is more widely available to patients without the need to create new facilities, while also benefiting the facilities themselves by improving workload management.